Hello everyone, welcome back to another educational video on Form 4 Biology. My name is Janet and I'll be teaching you with clarity whilst employing creativity and critical thinking to enhance learning. To receive notification of new biology videos, please click the subscribe button below to subscribe. Our topic for discussion today is Form 4 Biology, Chapter 4, entitled Chemical Composition in a Cell, Subtopic 4.2 on Carbohydrates. So the star of this lesson are the carbohydrates, led by Chef Potato. Here, what do dishes like nasi lemak, spaghetti, char kway teow, and roti canai all have in common as their main ingredient? Yes, it's the organic compound called carbohydrates, which we eat every day to provide energy for daily activities. So here are a few common foods with lots of carbohydrates in them. We have nasi lemak and potatoes, corn or maize, which we call jagung, spaghetti, sweet potatoes, wholemeal bread, and bananas. Let's look at an overview on carbohydrates. Carbohydrates can be divided into three types, namely monosaccharides, disaccharides, and polysaccharides. The word mono means one. So monosaccharides are the basic units of all the carbohydrates, and they are the simplest type of carbohydrates. They are also called simple sugars because they taste sweet, they are soluble in water, and can be crystallized. The chemical formula for all monosaccharides is C6H12O6. Examples of monosaccharides are glucose, fructose, and galactose. So in this video, we'll be studying monosaccharides, and disaccharides and polysaccharides will be studied in part 2, the next video. Let's look at disaccharides. The word di here means two. So disaccharides are formed when two units of monosaccharides join together in the process of condensation. Disaccharides are called complex sugars, and they have the similar properties of simple sugars. The properties are similar to simple sugars in that they are soluble, they taste sweet, and also can be crystallized. Their general chemical formula is C12H22O11. Examples of disaccharides are lactose, maltose, and sucrose. Polysaccharides, the word poly means many. So they are made up of many units of monosaccharides that are joined together through the process of condensation. Polysaccharides are called complex carbohydrates, and they have the general formula C6H10O5 bracket N. That means this unit of C6H10O5 is repeated over and over again, and this unit is joined. These units are all joined together, and the number of these units is N, which can be more than 100. So more than 100 of all these units joined together in a long chain. That's the structure of polysaccharides. Examples of polysaccharides are glycogen, starch, and cellulose. So in this video, we'll discuss monosaccharides, and in the next video, we'll be discussing disaccharides and polysaccharides. The learning outcomes or objectives are as follows. After this lesson, you should be able to list the elements of carbohydrates, state the ratio of hydrogen to oxygen in carbohydrates, explain the types of monosaccharides, their sources, physical properties, and general chemical formula. Explain the meaning of reducing sugars. There are two types of compounds in the human body. The first type are the organic compounds, and the second type are the inorganic compounds. Organic compounds contain the element carbon. That's the definition for organic compounds. But inorganic compounds do not contain the element carbon. The word in here means not organic. Inorganic or not organic. Organic compounds are actually produced by living organisms. For example, carbohydrates, proteins, and lipids are produced by plants and also by humans and animals. Inorganic compounds are usually not produced by the living cells, but they are obtained from the non-living external environment. For example, water. Organic compounds are more complex in structure, for example, proteins, but inorganic compounds are simpler in structure. For example, water again, or mineral salts like sodium chloride. Examples of organic compounds which we will study in this chapter 4 are carbohydrates, proteins, lipids, and nucleic acids. Examples of inorganic compounds are water, acids, bases, mineral salts, such as sodium chloride. They all do not contain carbon. How are complex organic compounds formed? Some complex organic compounds are formed from small molecules called monomers, which are the basic units or building blocks that combine together through condensation, a special process called condensation, to form the complex organic compounds or polymers. From monomers, we get polymers. As an example, let's look at this picture here. So all these small molecules are the monomers, the basic units. So when they join together through condensation repeatedly, many of the small monomers join together, they form a large complex molecule or macromolecule, a big one, called the polymer. Poly means many and mono means one. So polymers are formed from basic units called monomers through the process of condensation. What is condensation? Say with me, condensation. Condensation is a chemical process in which smaller molecules join together to form a more complex and larger molecule or compound with the removal of water molecules. Okay, for example, glucose is a monomer. So many molecules of glucose combine together through condensation to form starch, which is the complex organic compound, through the process of condensation. Now in this process, when the glucose molecules join together, water molecules are removed. They are formed, right? And that is what is meant by condensation. The opposite of condensation is hydrolysis, or the reverse process, whereby large organic compounds can be broken down into their basic units called the monomers with the addition of water, when water is added in the reaction. Now we'll be studying more of hydrolysis and condensation in the up and coming videos, so do not worry about it in this video, because we are going to discuss monosaccharides only for this video. Okay, so we discuss more of condensation and hydrolysis later on with clear examples for you to understand. So organic compounds are compounds that contain carbon, and they are complex compounds which consist of macromolecules, macro means large, very large molecules, that are usually polymers. Not all are polymers. Not all complex organic compounds are polymers. Some of them are. So they are formed from monomers. An example of these polymers are carbohydrates, proteins, and nucleic acids. Carbohydrates are the first organic compound that we will study here. Carbohydrates are eaten every day, usually with every meal, and we have found various ways to cook them so that we can eat them and let them provide energy for us to carry out daily activities and for our cells to carry out cellular activities. So what is their chemical composition? All carbohydrates contain the three elements carbon, hydrogen, and oxygen. The symbols are C, H, and O. So just remember CHO for the three elements in carbohydrates. Second point to note is that in one molecule of carbohydrate, the ratio of hydrogen atoms to oxygen atoms is always 2:1. For example, glucose. The formula of glucose is C6H12O6. H12O6. So there are 12 hydrogen atoms in glucose and 6 oxygen atoms in glucose. Thus, the ratio is 2:1. However, it is wrong to say that the ratio of carbon atoms to hydrogen atoms to oxygen atoms is 1:1. Okay, so the ratio of carbon to hydrogen to oxygen atoms in some books is stated as 1:2:1. That is not true for disaccharides and polysaccharides, although it may be true for monosaccharides. We'll talk about that later in the next video. Now, the general function of carbohydrates is to provide energy for daily activities and for cellular activities. So there are three types of carbohydrates that we must study. That is the monosaccharides, the disaccharides, and the polysaccharides. We'll start with the monosaccharides. The first type of carbohydrates are the monosaccharides. Let's say this word, monosaccharides. They are the monomers or basic units of carbohydrates and the simplest type of carbohydrates. So they are called simple sugars. Their physical properties are that they taste sweet, they are soluble in water, and they form crystals. All of them have the chemical formula C6H12O6, in which the ratio of the hydrogen atoms to the oxygen atoms is 2:1. There are three examples that we have to know, which are monosaccharides, and that is glucose, fructose, and galactose. They are all reducing sugars. We will discuss this later. And normally, monosaccharides are denoted by the hexagonal symbol, like this. This is very true for glucose and galactose, which forms a 6-member ring. Okay, so many students ask me, if they all have the same chemical formula, what is different? What is the difference between glucose, fructose, and galactose? So here I'll just like to go a bit further into the molecular structure of glucose, fructose, and galactose, which forms rings. And there's no need to memorize these diagrams actually, these three diagrams here, although you need to know the basic molecular formula of monosaccharides is C6H12O6. But let's have a look at this molecular structure shown here. So the difference between glucose, fructose, and galactose is in the arrangement of the atoms, and it's caused the structural difference in the molecule. They have different arrangement of atoms. For example, in glucose, we see that it has 6 atoms in the ring here. Now the carbon atom is not written down here. So actually every corner here has carbon atoms. C here, 1 carbon atom, 2 carbon atoms, 3, 4, 5 carbon atoms, and joined to 1 oxygen atom. to form a hexagonal ring. This is same also for galactose. But for fructose, it has a 5-sided ring or a pentagon shape. So the structure is different from glucose. And then the arrangement of molecules are different. For example, if you compare glucose and galactose, here you have for galactose OH or hydroxyl group on the top here, and the H is at the bottom. That's the reverse for glucose. H is on top and OH is at the bottom. So there are differences, but we do not need to know the differences specifically. Just to know that the atoms are arranged differently to form different structures in the molecules. Okay? That's all. But the facts on this side, on your left-hand side, they are all important facts to remember for objective questions, for example. What are the sources of glucose, fructose, and galactose in the food that we eat? Glucose is the most common monosaccharide, and it is usually used to build all the polysaccharides, most of the polysaccharides. Glucose can be obtained from rice. Actually, rice is starch, but starch is made up of glucose. So when it's digested in the body, the starch will become glucose because it's made of glucose monomers. Wheat. Now grapes have the glucose in them. Grapes have a high percentage of glucose in them. They are very sweet. Oranges and also bananas have a lot of glucose in them. For fructose is found in honey and sweet fruits like bananas again. Mango has a lot of fructose, and also apples. Some of the fruits we eat have a high content of glucose and fructose. Galactose is especially found in milk. It is a simple sugar that's found in milk. Now, earlier we mentioned that all monosaccharides are reducing agents. So what is reducing agent? What is a reducing agent? Or what is reduction, first of all? So we have to study some chemistry here in order to understand this word reduction and reducing agent. Now, reduction is a chemical reaction in which one of the reactants, let's name it A, gains electrons, E minus electrons. So when A gains electrons, we say that it undergoes reduction or is reduced because its oxidation state is reduced. So oxidation state is a term from chemistry, and we are going to explain this in the next part here. Okay, so example, reduction occurs when copper 2 ion Cu2 plus gains an electron to become copper 1 ion Cu plus. Okay, Cu2 plus E minus gives you Cu plus, and this process is called reduction. We say that the copper 2 ion is reduced to copper 1 ion, and the oxidation state has been reduced because this electron, which is negatively charged, neutralizes one of the positive charges on the copper ion. So from 2 plus, with one electron added to it, it becomes plus. One positive charge has been neutralized. So the oxidation state of Cu2 plus is plus 2, and the oxidation state of Cu plus is plus 1. So there's a reduction in the oxidation state, and we say that this process is reduction, and copper 2 ion has been reduced to copper 1 ion, Cu2 plus to Cu plus. Okay. Now the other reactant B involved in the reaction that gives this electron is called the reducing agent. So in this context, our reducing agent is actually the monosaccharide that donates the electron for the process of reduction. Let's go further and discuss the reducing sugars, the monosaccharides as reducing sugars. Let's explain a good question here, an important question. Explain why monosaccharides are called reducing sugars. 4 marks. Firstly, we have to mention that monosaccharides, all of them, act as reducing agents from the viewpoint of chemistry. And then elaborate what we mean by reducing agents. The monosaccharides can transfer or donate electrons or hydrogen to another compound and cause reduction to occur. So they can transfer electrons or donate electrons to another compound. Then we give the example of the Benedict's test. Thus, when a monosaccharide or reducing sugar is heated with Benedict's solution, which contains the blue copper 2 sulfate, monosaccharides will then reduce the blue copper 2 ions, Cu2+, to copper 1 ions, Cu+. So this is the equation for it. Cu2+ plus an electron from the monosaccharide becomes Cu+. And this is a reduction process, so that's why monosaccharides are called reducing sugars. Okay, this is the basic equation for the reaction between monosaccharides and the Benedict's solution. So now we have from copper 2 plus we get copper plus. What happens to the copper plus ion? So copper 1 ions or Cu plus then form an insoluble brick red precipitate of copper 1 oxide, which is insoluble in water, so it precipitates out as a solid. And this is what we see in the test tube after heating the mixture of reducing sugar and Benedict's solution. We get the brick red precipitate of copper 1 oxide. So here's a schematic diagram. It's not an equation, just to show you that copper 2 sulfate plus reducing sugar produces the brick red precipitate at the end of the reaction. So this makes the Benedict's test a test for the presence of reducing sugars, such as the monosaccharides. Let's look at the test called the Benedict's test for reducing sugars. What are the steps or procedure involved? Firstly, add 2 ml Benedict's solution to 2 ml of the sample solution that we are testing in a test tube. Okay. Next, heat the test tube in a boiling water bath for a few minutes. And if reducing sugar is present, the solution will change color from blue to green to yellow to orange, and finally a brick red precipitate is formed. That is, if reducing sugar is present. If the reducing sugar is absent, that means the solution will not change color. This is a negative test, and it will remain light blue in color. Let's look at the color changes in the Benedict's test for reducing sugars. If we were to mix some reducing sugars with the Benedict's solution in the test tube and heat it up, at first we will see that the color of the solution is blue due to the Benedict's solution, due to the copper sulfate in the Benedict's solution. As we heat it up, it will change from green to yellow to orange to brick red. Alright, so all these are positive tests for the presence of reducing sugars. But if it is just green or yellow, it shows less reducing sugar. Okay, and it also depends on the type of reducing sugar that's being tested. But all these are positive tests for presence of reducing sugars. Although the brick red precipitate shows that there's a lot of reducing sugar here, and it is the standard observation for the presence of reducing sugars. It's the positive test for the presence of reducing sugars in a sample of food. What is the use of Benedict's test? So the first reason for the use of Benedict's test, or the first use, is to test for the presence of reducing sugars such as fructose, glucose, galactose, which are monosaccharides, and maltose and lactose, which are disaccharides. So these five reducing sugars must be memorized, and they will give a positive result for Benedict's test, because they will all produce the brick precipitate when they are heated with Benedict's solution. Number 2, Benedict's test can be used to test for the presence of glucose in the urine of a patient or of a person to determine if the person suffers from diabetes mellitus or penyakit kencing manis. So diabetes mellitus is a disease where the person has an excessively high level of glucose in the blood, and thus the glucose is excreted in the urine. So for normal people, their urine does not contain any glucose. But for a person that's suffering from diabetes mellitus, his urine will contain glucose. So to test whether the person is suffering from diabetes mellitus or has glucose in the urine can be tested for the presence of glucose or presence of reducing sugars using the Benedict's test. So this is the use of the Benedict's test, especially for exam questions. If they ask you, how do we test for the presence of glucose, let's say, in the urine, then you can suggest using the Benedict's test, okay, to test for the presence of glucose in the urine, the glucose or reducing sugar in the urine. However, for doctors today, or even for patients who like to test for the presence of glucose in their urine, normally they will use a small dipstick with chemicals to measure the glucose levels in the urine of the patients. So this is a commercially prepared product where inside the bottle you get this dipstick, which are pieces of cardboard or paper that have special chemicals on them to detect the presence of glucose in urine. So these strips are just dipped into the urine samples, and the color changes will tell the doctor whether there is any glucose in the urine. You will be able to measure the levels of glucose in the urine. So this is just an interesting fact for you to know. Here is a good memory aid for you to remember the 3 monosaccharides: glucose, fructose, and galactose. Remember, glum, fruit, gal. Instead of girl, we can call this girl's gal. Glum, fruit, gal. Glum is for glucose, the first 3 letters. FRU is for fructose, and GAL is for galactose. So maybe this girl, she was selling some fruits like apples, and nobody wanted to buy her fruits, or very few people bought her fruits, so she's glum. Glum means to be very sad. Glum, fruit, gal. Okay, this is a good memory aid, and I'll be giving you more memory aids for the disaccharides and the polysaccharides in the next video. That's all for this lesson. In the next lesson, we will continue to study carbohydrates. It will be part 2, and we'll be studying the disaccharides and the polysaccharides. And I'll give you another memory aid to help you to remember the disaccharides and the polysaccharides. Thanks for watching. Stay tuned for the next video. Like, comment, subscribe, and share to your friends. You will find this helpful. Especially during this time when you are staying at home, make good use of the time and watch the videos, as this is like a normal lesson in class, and you will benefit from it, especially because I've given notes that are more detailed to help you learn. I'll be giving another series of videos later on. I'll be producing another series of videos for revision. That will be shorter and more compact. For now, these are all teaching videos to help you understand better the topics that are being taught in school.